In the next 20 minutes or so, I'm going to try and give an overview of what it is we do in the biotechnology development group. And for this, I'm going to be splitting the talk basically into two sections. The first regards what we do regarding facilitating the access to quality and affordable biotherapeutics. And this paragraph here basically explains why we're doing this. The first sentence is it's basically because no one can deny the tremendous health benefits of biological drugs. And I always use the example of insulin to emphasize this point. Insulin was the first recombinant drug to achieve FDA approval nearly 25 years ago now. Before that, it was being produced from pig parts, pig pancreases, and it takes approximately 2 tons of pancreas to produce around 150,000 doses. Today, using recombinant E. coli expressing insulin with a 500-liter fermenter, we can produce basically the equivalent amount of doses. And this is extremely important if we consider the number of people that are suffering from diabetes worldwide. These numbers are taken from the World Health Organization website. You can see that approximately 350 million people suffer from diabetes. To put that number in perspective, that's basically the population of the United States with Italy thrown in. This number is going to increase. It's projected to increase over the next few years. In 2030, the WHO projects that it's going to be the 7th cause of death. And similar to cardiological problems, also, again, 80% of the people with diabetes will live in low and middle income countries. And I think this number is basically always going to be the same because 80% of the people in the world live in low and middle income countries. But basically, this point allows me to emphasize the second reason we're trying to facilitate access to biotherapeutics. And that's they're unaffordable to the majority of people on this earth. And in countries where they are available, they do represent a significant burden on the healthcare system. This is always an example of insulin. In the UK, the National Health Service budget, basically 10% of it is spent on curing diabetes, and 8% is basically just for the insulin necessary. So that's basically 1% of the UK National Health Service budget is spent on the drug itself. Going to Italy, just to give you an idea of how costly biological drugs are, we spend basically in 2012, we spent 3.5 billion on biologicals. That's 12% of all the money that's been spent on pharmaceuticals. And if we consider the pharmaceuticals that are reimbursed by the healthcare of the Italian government, that number increases to 34.5%. Now, loyally, these biologicals are now off patent, and a lot more are going to come off patent. This provides the possibility to produce them as biosimilars and reduce expenditure. And I've just shown an example here of filgrastim, Gcsf, and the biosimilar basically of Neupogen. It's one of the first biosimilars approved in Europe, and since its entry into the market, you can see that it's resulted in a great price variability in Europe, but basically on average, the biosimilar is around 35% cheaper than the originator, and it does even reach peaks of around 80%, for example, in Slovenia, which is neighboring just across the border here. Not only do biosimilars reduce the cost, they also increase the accessibility of the patient to the drug. In the UK, again, the entry of Gcsf into the market increased patient access by 50%, and this was associated with reduced readmission rates. So, I've explained why we're concentrating on biotherapeutics and biologicals. How are we going about it? Well, the idea around 10 years ago now was if we could create a facility at ICGEB that could provide support to local biopharmaceutical manufacturing in member countries. We could provide cell lines, protocols, standard operating procedure, analytical methods, and more important, reference materials, and more importantly of all, training of the scientists from the member countries at ICGEB in these technologies. This has been an ongoing process. I remember 10 years ago with the head of the group then, which was called Sergio Tismineschi, we started off with these small 4 centimeter columns. Now we're on to meter high columns. We also continuously upgrading the technology, looking for methods to improve it, to make it more efficient regarding the cost of goods. For example, this is a publication one of our first PhD students did in 2012, where we were looking at the role, we were trying to increase the yield of the CHO cells we have producing erythropoietin, and we were using dextran sulfate. And basically, we found that due to its anti-apoptotic and pro-autophagia properties, we were able to increase the yield of erythropoietin production twofold. And here, basically, you can see, I think it's useless to look at all the gels, but you can see just by eye that the cells on the 5th day of production are much more healthier when the dextran sulfate is added to the media rather than without. We also concentrate a great deal on the quality, because there's a big, there's lots of discussions on the quality of biosimilars. We follow the European Pharmacopoeia monographs for all the biologicals we produce here. Step by step, we've developed all the methodologies necessary or requested by European Pharmacopoeia to analyze the protein. So it's not just a question of production and purification, but also of quality. Here I've shown again erythropoietin because it's the most, well, it's one because it's nicer gels. Erythropoietin, as you all know, is a glycoprotein. It has several different isoforms with different glycosylations. This gel here basically shows, this lane here is our supernatant attained from the cells. Then we do an affinity purification. We do an anion exchange, a cation exchange, and this is our result. Basically, these isoforms are identical to that of the European standard and fall into what's requested by European Pharmacopoeia, so which allows a certain percentage of each isoform to be present in the final drug, in the final protein. We also analyze this by isoform distribution, and this isoform distribution by capillary zone electrophoresis, which is shown here. Again, this is the standard. This is our protein, and we also perform or develop methods for the biological assays. In this case, basically, it's an in vivo assay just done to perform, done to measure the increase in reticulocyte production in mice. Oops. So that was just an overview of what we do, but this is basically the portfolio we now have in place. These are the biologicals we can produce here at ICGEB. This is basically the batch sizes where we are now for these proteins, and here it's in grams, and here you can see the batch size in number of doses. You'll notice that in most cases they are at pilot scale. Obviously, some procedures need to be scaled up. For example, production of a PEG EPO, where quite low amounts. But I use this slide to highlight that if we compare the batch sizes that we can produce here at ICGEB to what's used in a country. So if we take interferon alpha 2A, the batch size we produce now is around 250,000 doses. So in Italy, we use around 4 million doses a year. So it can be easily, how can I say, transformed in a member country to the amount needed to cover their market. For example, in Egypt, I saw that there's around 500,000 doses used a year. So basically 2 batches of what we can teach would cover the Egyptian market. We also performed, and I forgot to mention this, the chemical modification, the pegylation of some of these biologicals. This is the new generation, basically, with just the covalent attachment of a polyethylene glycol. It makes the biological less immunogenic and reduces and improves the pharmacodynamics and kinetics of the biological in the bloodstream. Our chemistry group has been able to do that. Again, Let me just go through this. The batch size we can transfer of pegylated interferon alpha 2A is around 8,000. Not enough to cover, let's say, the doses necessary to cover Italy, but basically 12 batches of this would cover the doses necessary to fulfill the requirements of a market such as Iran. And Iran. So the procedure for a tech transfer at ICGEB basically foresees the finalization of a technology transfer agreement between the interested party and ICGEB Trieste. It usually provides, well, it always provides for a period of training in our laboratories here of the industrial partners' employees. Normally, 4 employees come and spend between 4 to 6 weeks in Trieste. They first of all observe us perform a full batch, and then subsequently they perform it themselves with us watching over them. And we also provide assistance in many cases, either electronically or also in the interested party's own premises. This experiment, because it started out as an experiment, has been extremely successful. We've signed more than 41 collaboration agreements in the last 10 years. These agreements have allowed us to train around 83 scientists in our laboratories and have been signed in 22 different countries that you can see here in this world map. The countries in yellow are the ones which we have transferred the technology to. The countries in green are the ones where I know that the companies have arrived to the market. Maybe there's more, but I haven't been able to follow all of them. For example, in Argentina, with our technologies, EPO production has been on the market for at least 10 years. Something that happened last year, which made me really happy, was one of the first companies that we transferred Gcsf to, which was called Int'l. For many years, they were producing Gcsf in India, and last year they applied for EMEA, European Medicines Agency approval, for now to be able to now sell the Gcsf in Europe, and they actually obtained it. And this was, when I was reading through their documentation, it was exactly the same procedures we are still transferring today. No modification was done on it. And that basically covers the first half of the talk, the text transfer. I just wanted to mention, and this is very, very briefly, another area we're trying to start working now, and that's more translational biology, trying to bridge the gap between academia and the big pharma. And as Professor Jack has said this morning, the big pharma are always asking academia to take a few steps further towards them before actually investing money in an idea. You heard earlier from Professor Baralle something about ALS, amyotrophic lateral sclerosis. This is what we decided to concentrate on. It's a motor neuron disease. It has a late to mid onset in life. It affects around 2 in 100,000 people. And once you're diagnosed, life expectancy is between 2 to 5 years, from the time of 2 to 5 years. He also showed you that, well, in 95% of the cases of ALS, these are characterized by these large inclusions of TDP43. And he also mentioned that, well, I'm not sure if he mentioned it. There's a big debate going on. Are these large inclusions toxic per se? Or, as we like to believe in the lab, is the fact that the inclusions, the aggregates in the cytoplasm, sequester or the TDP43 in the cell, resulting in a loss of function, the cause of the disease? Irrespective of whatever pathway is true, loss of function or toxicity, it's logical that if we reduce the aggregates, it may be beneficial for the patient, either by taking out the toxicity or by not sequestering the newly synthesized TDP43. So we decided to create some TDP43. So we decided to look for, to create assays to look for compounds that could enhance aggregate degradation, and that can occur by two principal paths, either autophagy or UPS degradation. And we started developing assays to see, to be able to perform a drug screen. The first assay we developed was basically a cell line, of which we wanted to use to perform a high throughput screen. This cell line creates aggregates, and these aggregates are created because we took a prior on light domain of the TDP43 in the C-terminal tail and added 12 repeats of this to GFP. And when we transfected the cells, we can see it results in aggregates. Not only does it result in aggregates, these aggregates, and this is a stain for TDP43, sequester endogenous TDP43. So this, to us, was a good model to which perform the high throughput screen. Before performing the high throughput screen, we wanted to set up a panel of compounds already tested to be able to clear the aggregates. So we took several drugs known for their ability to enhance UPS or autophagy and performed a clearance assay. Now, the clearance assay is very simple. Basically, we induce the aggregates with tetracycline, because I didn't mention, but these cell lines, when we perform the transfection, it's an inducible system. We then stop induction by washing the aggregates, add our compound, perform cell lysis, and look at the amount of GFP produced. And you can see, for example, and this is a case of Nortriptyline, one of the compounds we tested, that when we add this lane here, so when we add Nortriptyline, we have a reduction in the GFP. So it's basically either enhancing the degradation of the aggregates. We also found another positive compound, which was thyroridazine. And we keep on looking. We followed up the mechanism of action of Nortriptyline to see how it was increasing degradation by looking at a few classical markers. You heard this morning LC31 to LC32 convergence as a marker for autophagy. We saw that Nortriptyline was increasing autophagy. P62 levels are also a marker for autophagy, or rather the marker for lysosome degradation. However, in this case, we didn't see a reduction of P62, but an increase. So this highlighted the fact that it wasn't really, notwithstanding Nortriptyline was increasing the levels of LC3 and pointed towards autophagy, it wasn't really the degradation of the aggregates wasn't really occurring through autophagy. And this was also highlighted by the fact if we block autophagosome and lysosome fusion with ammonium chloride, we can still see the reduction in the aggregates. So then we went on to the obvious alternative, and that was the UPS pathway. And here, if we block the UPS pathway with lactacicine, we can see we block the degradation of the GFP12QN aggregates. These few preliminary experiments were enough to obtain a grant from Alzheimer's Research UK. We're now performing with these cell lines and in collaboration with the MRC Technology Group in England, a preliminary screen using 10,000 compounds to test for chemical compounds that are going to be able to induce this aggregate degradation. This has just started. There's a lot of work to be performed in the development of these assays. The top hits will be tested in vivo. We're creating a mice model underway, and we already have a fly model. This fly model is basically made by expressing underneath a neuronal promoter in the flies the GFP12QN aggregates, and they do result in a phenotype. You can see basically that the flies expressing the GFP12QN have problems in mobility. Well, you should be able to see it. Whereas the wild-type flies climb easily. Just to get the assays in place and ready for the high compound screen, we've also tested Nortriptyline in the flies, feed it to them, and you can see that, so basically this is a climbing assay of flies not treated. these are the flies that express the GFP12QN, and these are the flies that have been fed nortriptyline, and we can see that basically the difference in the climine between the flies expressing just GFP and GFP2QN disappears when we treat them with nortriptyline, improving the phenotype. But these are very preliminary experiments that need to be repeated. Basically, at the end of this screen, we hope to be able to offer the farmers basically a platform that has been at least originally tested, and for them to carry on the work in the drug discovery. So that's the end. I just want to highlight the people that do the work. This is basically the biotechnology transfer group. These are the chemists here. Natasha, Sulena, and Vensi are basically perform the protein purification, the production of the cell lines. The work on TDP-43 and the drug screen is done in collaboration with Professor Baralle, Emanuele Borati, and a lot of the work was done by Lara De Conti and Lucia. And thank you for your attention.